Sorry, I forgot to hit the record button. Nothing we do here would be possible without their support. So thank you for supporting us. We want to remind you once again to save the date for our Tick Academy on October 16th and 17th. If you have not been before, this is a great opportunity to learn a whole bunch of things in just 1 or 2 days. It's 2 half-day webinars. We bring in 5 or 6 speakers each day. There'll be some panel discussions and all kinds of fun stuff. And we look forward to seeing you there. Registration will be live very soon. We are working on it. Apologize for the delay on that, but it will be coming soon. So keep an eye out for that. Next month's webinar will be same time, same place. So July 10th, 1 p.m. Central Time, second Wednesday of the month. And that'll be Dr. Roger Lane from LSU, and he will present on historical and ongoing nucleosome research. They just received a big National Science Foundation grant over there to do some cool research on with nucleosome, and he's been involved with the product for a long time, so he'll have quite a bit to share with us. So put that on your calendar if it isn't already. As always, please contact me if you or someone you know would like to present at a future meeting. And my email address is right here. You can also reach me through Basecamp. Do we have any updates for the working group? It looks like Monica has one. It says the next meeting of the TBD and Associated Illness National Community Engagement Initiative led by HHS will be early 2025 in Portland, Maine. So keep an eye out for more details on that. Monica, is there anything else you'd like to share about that?
Only that they held their first meeting yesterday in California and virtually as a kickoff to this, public engagement, continuing public engagement regarding these issues. So the Portland date will be their second session. So yeah.
It's good. Thank you for sharing that. Anything else? Anyone else have anything they want to share? If not, we'll keep on going. So today's presentation will be from Dr. Nicole Chinnici, who's the director of the Dr. Jane Huffman Wildlife Genetics Institute at East Stroudsburg University, where she's focused her research on ticks and the pathogens they transmit. Dr. Chinnici pursued advanced studies in epidemiology, where she delved deeper into the complex ecology of ticks and the pathogens they carry. Her work spans fields such as microbiology, epidemiology, and molecular genetics, with an emphasis on tackling this global health challenge. Under Dr. Chinnici's directive, the Tick Research Laboratory was established in 2018 to evaluate the dynamics of tick populations and the factors influencing the spread of tick-borne pathogens. Findings of the research will contribute to the development of innovative strategies for disease surveillance, risk assessment, and vector control, enhancing our ability to combat emerging threats. Beyond her scholarly pursuits, Dr. Chinnici is deeply engaged in outreach and education efforts aimed at raising awareness about tick-borne illness and promoting preventative measures. She is a devoted advocate for public health initiatives and policy measures aimed at mitigating the impact of these diseases on both human and animal populations. Do you have a quiz question? Stop sharing and add and watch the quiz question. Can everyone see this on their screen? Looks like people are answering. Perfect. And while you are finishing up the quiz question, I will ask Dr. Chinnici to please share her screen. Everyone, please welcome Dr. Chinnici. Thank you for being here.
All right, thank you, everyone. Good afternoon. Let's see here. All right, so I'm Dr. Nicole Chinnici. I'm the director of the Dr. Jane Hoffman Wildlife Genetics Institute. We're part of East Stroudsburg University, and we house the Pennsylvania Tick Research Lab. We've been doing tick research since the early 2000s, but we established the research center specifically focused on ticks in about 2018. So today I'm going to spend some time just really talking a little overview about some of the research that we're doing, a little introduction about our lab, and then go into some passive and active surveillance. I know a few years ago I presented on our passive surveillance, so I'm really going to focus more so on the active side of things and some of the mitigation strategies that we're starting to implement and evaluate in the field. So a little bit about our facility. We're located out of the Innovation Center of East Stroudsburg University. We have about 16 staff members ranging from both field and laboratory technicians and research assistants. And then we have anywhere between 10 and 20 research students, whether they're graduate or undergraduate students working in our institute on small research projects, as well as on our larger research projects that we are conducting. We have several lab spaces, all designated to different aspects of the molecular technology. So we do a lot of molecular detection of pathogens in ticks, in addition to some of these other methods that we're doing. And we talk a little bit about why Pennsylvania. So just so I know everyone's all over the place, a lot of the diseases associated with ticks are concentrated in the upper Midwest and the Northeast region. When we zoom into that Northeast region, Pennsylvania has a lot of cases of reported tick-borne illnesses, and we're one of the leading states for those cases. So we really wanted to dig deeper into what's happening at the tick level as well as the human level. So we've received funding to do some passive surveillance, and passive surveillance is essentially just requesting the residents of Pennsylvania to submit their ticks into our facility. And if they are a resident and they have a physical address in the state of Pennsylvania, then they qualify for free tick testing. The ticks are mailed to us or dropped off at our facility, and we conduct qPCR testing for 5 different tick-borne pathogens. And what we're doing with that data is we are really compiling a twofold analysis with it. We are collecting some information of tick-borne diseases in Pennsylvania, looking at some hotspots and what individuals are being exposed to, looking at the distribution of ticks and what the human and domestic animals, what that population looks like for exposure, and some demographics on tick bites. So we request information of like where the tick was feeding on the person, attached to skin, where they feel they may have been exposed, information such as that. And then in a flip, it allows the individual to identify if they've been exposed to a tick-borne illness, and it also identifies their risk. So we'll look at how engorged that tick is, and then they can use that information with their physician to better understand if symptoms arrive or what some type of diagnostic testing that they need to undergo. The different pathogens that we do test for the passive surveillance, if it's a black-legged ixodes tick, it's tested for Lyme disease, anaplasmosis. We differentiate between variants on anaplasmosis to determine if it's human versus deer variant. We're looking for tick-borne relapsing fever, Borrelia myomodi, Babesia microti, and Powassan virus. And if it's a non-black-legged tick, a dermacentor or amblyoma, we're looking for Lyme disease, ehrlichiosis, specifically Ehrlichia trephiensis, Rocky Mountain spotted fever, Rickettsia rickettsii, tularemia, as well as Powassan virus. I'll go into a little bit of our results. So in last year, just a little summary of what we looked at. So we had over 18,000 ticks submitted for testing. Of those, the majority of them were black-legged ticks. We had 8,600 adult black-legged ticks and just under 4,900 nymph black-legged ticks submitted for testing. When we looked at how many were infected, 50.6% of the adults were infected with at least a single tick-borne pathogen. The majority of that being Lyme disease, but we also see high rates of anaplasmosis and other tick-borne illnesses. We saw our state co-infection rate in general was 8.39% in the adults, but it ranged upwards of 10.6% in certain regions. So there is some regional differences. For the nymphs, a third of them tested positive for a single tick-borne illness. 9% of them being co-infected, ranging as high as 5.5% infected. Again, with some regional differences. We saw in all regions of Pennsylvania, so this map is just taking Pennsylvania and breaking it down into some 6 regions. We found all the tick-borne illnesses, so Lyme disease, anaplasmosis. This is again focusing on Ixodes scapularis. So Lyme disease, anaplasmosis, Babesia microti, Borrelia miyamotoi, and Powassan virus in all of the regions. The only regions we did not detect Powassan virus was in the nymph population in the north central and the south central regions. When we looked at the co-occurrence of tick-borne illnesses, so this is looking at how these co-infections are circulating in the environment. We have adults and nymphs on these heat maps. So this is looking at whether or not the prevalence of having more than one tick-borne illness is greater than the expected. Based off the individual pathogen prevalence. And we see that these diseases are positively associated with being co-occurring in a tick population than they would be at a single infection for most of the tick-borne illnesses. So these little nomenclatures here, so BBU is Borrelia burgdorferi and Babesia microti, Borrelia burgdorferi, Anaplasma phagocytophilum, and then Anaplasma phagocytophilum with all 3 co-infected had a positive correlation. And then the same we saw with the nymphs, seeing a positive correlation with Lyme disease and Anaplasma phagocytophilum, Lyme disease, and Powassan virus, and Babesia microti with Lyme disease and Anaplasma phagocytophilum. So we're seeing this positive correlation that these diseases are occurring more frequently than they are expected based off their individual prevalence rate, and that's just in the passive surveillance that we're seeing some of this data. We've been really diving in and compiling this data to look at it a little bit more specifically, but we've been able to map out some of the peak seasons of our tick life cycle, which overlaps a little bit with how we're seeing the life cycles and the cases in the human population. So really looking at when these ticks are being submitted by individuals and understanding their exposure date so that we know when these ticks are becoming more common. We have seen the nymph population to start to emerge a little sooner. So we're starting to see nymphs in March that are being submitted for testing. And then the adult ticks seem to kind of stick around a little longer than in the past years. So we're taking into account with some other factors that may be associated with the risk of exposure to some of these ticks and the incidences of ticks, as well as the diseases in that passive surveillance aspect. We've quantified the regions where reported. So individuals, when they report exposure to tick bites, we ask them where they remove the tick from on their body. And specifically looking at the adult black-legged tick, the majority of removals were in the hairline and behind the ears. So us quantifying some of these exposures will better help us with targeting these educational programs to help individuals identify where the ticks could be attached and feeding. We've looked at where individuals report exposures, and we've seen that the greatest exposure to a tick bite has occurred in individuals' own backyards. A lot of times they're doing things such as playing with their children out back or doing yard work is the activity that they're reporting. Areas of the tick attachment we have seen associated with activity. So in general, when we're asking them to report where the tick was attached, as well as what activity they may have been conducting, we've seen an association, a positive association, where if they're doing anything like gardening or yard work where their hands may be in the area of exposure to the leaf litter or the ticks questing, the ticks did tend to attach on the waist and above on the body, whereas individuals reporting hiking, walking the dog, running, walking, things like that, the ticks were more associated with attaching from the waist down, so more on the leg regions. Our data showing that individuals ages 0 to 9 have the greatest risk of exposure to ticks, where these blue bars are the black-legged ticks and the red is the American dog tick. So the same thing we're seeing in human cases, the majority of tick exposures are in our children under the ages of 10, peaking again in the middle, like 35 to 39, and then in the older individuals. Looking at the length of time ticks are attached, we are better at identifying the adult ticks attaching pretty quickly due to their size, where on average it does take us around 18 hours to identify a nymph black-legged tick attached. So some of this is associated with better understanding what to look for and doing faster tick checks so that we can capture these ticks before they're attached and feeding. So then when we look at the broad scheme of things and why we're moving into some active surveillance, so we are really better understanding our exposure with our passive surveillance, getting an understanding of how individuals are exposed, what preventative measures they might be taking, what activities they might be doing when they're exposed. And now on the flip side, we're trying to look at what's driving these tick-borne illnesses in the environment. And looking at some different associations with the effects of these on the abundance of ticks in mammals, as well as the infections. So with our active surveillance project, we first started it last year, the fall of, I think it was 2022 was when we first launched in Northeast Pennsylvania, and spring of 2023 in Northwest Pennsylvania. So we're in 4 counties of Pennsylvania. We did our first year of baseline data, getting a better understanding of densities of ticks at all of our sites, density of small mammals, occupancy of large mammals, what large mammals are in the area, and what factors and roles they may play in the disease ecology cycle. Looking at the density of infections, some vegetation composition at each site as well, so we can better understand what might be driving these tick-borne illnesses and why there's differences in different regions. And I'll talk a little bit about that in a couple slides. So we are across 36 different sites in Pennsylvania, and the site setup looks a little bit like this. So we have 11 plots in various different areas. So we are in community parks, we're in state parks, we're in residential areas. And in those areas, we have 11 plots, each 750 meters square. And it's about a half-mile transect, and we tend to stick along the trail edges or the forest edges where we set up those plots. So this is an example of one of our sites at a community park. We have, here's an example of a plot. So within each plot, we have these subplots where we take some vegetative surveys to get a better understanding of the composition of the veg in that area. We have 10 meters into the trail edge is where we set up our trap line for small mammal trapping, and the small mammal trapping occurs every 10 meters along the transect of the entire plots. And then we also do our tick drags, where we drag 10 meters in and then 10 meters out of the wooded line to better understand densities of ticks. Our sites are located, there are 18 of them, 9 in Monroe County and 9 in Pike County, Pennsylvania. And then we have 9 in Mercer and 9 in Crawford County, Pennsylvania. So we have 2 different sides of the states purposely set up just as we're seeing in our disease surveillance. There are some differences in disease surveillance in these areas. We have different composition of vegetation in these areas. We have different elevation, so things look different. So we wanted to kind of have a separation of what factors might play a role when we have different environments associated with what we're evaluating. So when we're looking at our baseline, some of those environmental variables that we're collecting, we're looking at canopy cover per site. In addition to that, we're looking at the forest edge composition and the amount of forest edge that's present in these areas. We're looking at leaf litter death and how much leaf litter is present and how that might be associated with higher or lower densities of ticks. Looking at the vegetation composition, so quantifying grasses, forbs, bare ground rock, things such as that. Looking at slope, elevation aspect, anything that may play a role in the changes of the composition that may affect or promote the abundance of small mammals and ticks. We are collecting data at each site. So we're looking at the temperature, relative humidity at each one of these locations. We're also taking soil moisture levels, the day of tick drags, and various other different environmental factors that might play a role. At each site, we're doing collection of ticks. So we're looking at questing ticks collected using dragging methods. So those drag cloths, corduroy cloths, 1 meter by 1 meter square cloths. 10 meters are checked every 10 meters for adults and every 20 meters for nymphs. Those ticks are collected for disease testing. And then with our small mammal captures, we're also collecting any ticks that might be feeding on these small mammals during our trapping efforts to see the composition and diversity of ticks in these areas that are also feeding on our small mammals. When we're doing the small mammal trapping, we're also looking at small mammal densities. So we do 3 to 4 consecutive nights of trapping using Sherman traps. There's about 85 traps per site, depending on how loopy or windy the site is. When we handle new animals, all of them are, we take an ear punch sample, a blood sample, we collect ticks, and then each new individual is tagged with an RFID PIT tag, so that we can do some Jolly-Seaberg modeling, looking at densities of these small mammals. We're also quantifying large mammal occupancy. So at each one of our sites, we have 2 cameras that are set up each season. And during that season, they undergo a 15-day interval where they capture individuals moving in and out of these areas. We have scented and unscented cameras, so one's scented, one's unscented, and looking at the various different mammals that are coming into the area, looking at presence of mesocarnivores as well as the white-tailed deer occupancy to better understand whether or not large mammals have a impact on the changes of these environments. So we're still processing a lot of our samples, so I'll just go through some of our preliminary data just to kind of give you an idea of the massive amounts of samples that we're collecting in this project and some of the interesting findings that we're already starting to see. So in terms of ticks collected in our drag methods across all of our sites, Northeast is in the blue, Northwest Pennsylvania is in the gold, the number of ticks that we've quantified. So we've collected over 19,000 different ticks, and that doesn't include the quantification of any larvals that we collected. We just put the larvals aside for now, and we're focusing just on the adult and nymph ticks in those collections. Of those ticks, we have tested some of them, and I'll talk a little bit about them. But right now, for terms of like the majority, we're seeing mostly Ixodes scapularis amongst our sites. We've collected some dog ticks, but the majority of those dog ticks have been associated with Northeast Pennsylvania, and very few have been collected from Northwest Pennsylvania during these collections to date. We have had over 5,000 ticks identified on our small mammals. The small mammal diversity was much different. So we've collected other ticks outside of our tick drag. So we've seen Ixodes cookii, Ixodes angustus, some brown dog ticks and other Lone Star ticks in feeding on the small mammals, but not collected via tick drags. When we look at the density, so I highlighted this red box because we didn't have Northwest Pennsylvania data for the fall of 2022, we didn't get the sites up and running quite yet. So summer of 2023 and fall, we have direct comparisons between the 2 regions. In the summer, the nymphs are down here. We would suspect that the nymph densities would be higher than adults, which is what we see based off the life cycle. In terms of those densities, Crawford County had a higher density in comparison to the other areas. But in general, we had a pretty comparable density of nymphs in both Northeast and Northwest Pennsylvania. When we look at the fall densities, the adults should be more common, and that's what we're seeing here is our densities of adults are higher in the fall. The densities on the Northeast side of the state were much lower than the Northwest side of the state. So the Northwest had upwards of about 18 ticks per plot, where in Northeast Pennsylvania, we had about 4.5 ticks per plot. So much higher density in the adult ticks in Northwest Pennsylvania in comparison during that season. When we look at pathogen rates, we only have a proportion of this data. We're still analyzing all the data that we've collected so far. So we only really have Northeast Pennsylvania data, but of those ticks that we tested, we have found all of the tick-borne illnesses in adults. So Lyme disease on average was 47.5%. Monroe County was slightly higher than Pike County. We found anaplasmosis 6.9, 2.28%. Babesia microti was much higher in Monroe County at 11.9% and lower in Pike County. So there are county differences between this. I don't have our site-to-site variation, but we also saw within county variations of these tick-borne illnesses. And we found some pretty high co-infection rates as well. The nymph population, we identified all of the tick-borne illnesses except for deer tick virus, Powassan virus lineage 2. We did not identify in any of the nymphs that we have tested to date, but we are still going through that data. When we look at the population sizes of the small mammals, so we did have a diversity of small mammals that we collected. The diversity was higher. We had more diverse populations of small mammals in Northeast Pennsylvania. So we saw Peromyscus leucopus was the most abundant of all of the small mammals we collected. So we'll focus on that data, but we did have some Peromyscus maniculatus in Northeast Pennsylvania. We had some other shrews and voles as well collected, but the majority of Northwest Pennsylvania was Peromyscus leucopus and a few shrews. There wasn't as much diversity in that small mammal occupancy. So the estimate of the population based off mark recapture data, darker green being higher and red being lower. Our densities in Northwest Pennsylvania were much lower than Northeast Pennsylvania. However, we did have an issue with raccoon disruptions in our Northwest sites than we did in Northeast sites, and we've implemented some strategies to reduce that. So we do suspect potentially that could be a result of why we're seeing this result with the lower populations. When we look at the pathogens that are within these small mammals, we identified a much higher rate of tick-borne illnesses associated with our small mammals on the northeast portion of Pennsylvania. So we found Babesia microti, red. The size of the circle is sample size, and the color red is greater infections on these charts. So we had much higher infection rates in Northeast Pennsylvania compared to Northwest Pennsylvania. We only had, I think it was 1 or 2 small mammals infected at this one site, where we did not find Babesia microti in any of the other Northwest Pennsylvania sites in the small mammal population. And these are from blood samples specifically, I should mention. When we looked at Anaplasma phagocytophilum, we did see, again, a higher rate and more sites associated with the infection on the northeast side of the state than we did on northwestern side of the state in the small mammal population. Some of these areas over here that are white are higher elevation. So we're looking at the effects of elevation potentially on the influences that they may have on pathogen diversity as well as pathogen prevalence. We looked at some environmental influences on the population size in general. So looking at what might be influencing the differences in the size of the population of the leucopus mice, the white-footed mouse. And we found in Northeast Pennsylvania, when we looked at these variables like white-tailed deer occupancy, understory height, temperature, etc., we found relative humidity had a positive effect, elevation, and the proportion of Babesia microti infections. And then when we looked at both Northeast and Northwest Pennsylvania, that association with Babesia microti had a stronger effect on the size of the population. So we're further looking into potentially why Babesia microti may be associated with greater densities. Again, there we might have an influence of an issue because we had some disruptions from raccoons. So we're further diving into this data. So looking at what environmental factors might be driving this higher rates of Babesia microti infection in the mice. We found that in general, the relative humidity had a negative impact. So when relative humidity increased, Babesia microti infections decreased. We found the population size of the small mammals played a role, and the density of the tick species also. So the density of Ixodescapularis had a positive effect. And then looking at all regions, the population, the size of the small mammal population was definitely driving the most positive effect on the Babesia microti rates. When we looked at Anaplasma phagocytophilum, temperature played a role in Northeast Pennsylvania as potentially a positive association. And then when we looked at all regions, there was no factors associated with the presence of Anaplasma phagocytophilum. So now what we're doing is we're finishing up our analysis on the baseline. So we've captured baseline data, some of that I just presented, of what's happening in each of these sites and between sites. And now we're looking at implementing some mitigation strategies. So we're using a bioinsecticide, so Lalagard M52, which is MET52, which is a fungal spray that's targets specifically on ticks and mites. We're looking at a anti-tick and Lyme disease bait stations, the U.S. biologics bait stations with the Lyme disease and the anti-tick vaccines, and the combination of both the vaccine and the bioinsecticide, as well as some control sites, to see the effects that these methods have on essentially reducing the infections and densities of the ticks that we're seeing. So here's just, we did start our treatments this spring. Our bait stations are on their third cycle. You can see here a small mouse going underneath the station to access the vaccine, and then some other visitors interested, even though they have no sense. We had some bears come through the area to check out our stations. So for the future, we're still processing a lot of our samples. We are anticipating to have some of that done by the end of the summer to really have a better understanding of what's happening between these areas. We're looking at other environmental variables like atmospheric pressure, soil moisture, snow depth, lagged environmental variables, and other factors that might be influencing or driving densities of various different ticks, as well as the small mammals. We're accounting for differences in seasonality when it comes to recruitment and loss of our small mammal populations. We're looking at potentially adding on some new sites and new counties, as we're seeing some differences in the potential eastern to western spread of diseases. We're looking at improving our occupancy analysis and stimulations and better understanding how large mammals are going to play a role in this disease ecology cycle. And then starting to implement these tick mitigation strategies and then how they play a role and evaluate their effectiveness. So that is a grand overview. We are a pretty big team that we are focused on really driving deeper into these tick-borne illnesses in the state of Pennsylvania and how they differ between different regions of the state. Ticklab.org is our testing website. You can also visit there for more information. We're pretty active on social media, PK Ticklab. We thank the Department of Education, Department of Health for our funding for our research. And then if anybody has any questions.
Thank you so much for presenting today. We do have at least one question. They said, can you elaborate on raccoon disruption?
Yeah, so we had a handful of sites, not all of our sites, but there were a few sites in our northwestern Pennsylvania region where the raccoons would, when we would set our traps the next morning, come back to check our traps, raccoons pretty much disrupted anywhere between 50 and 80% of the traps, and pulling out the peanut butter and the fluff that we had in there. Sometimes they would even pull out the small mammals that were in the traps as well. So we've implemented some raccoon exclusion devices, and that has seemed to eliminate these raccoons from coming in and basically ransacking all of our traps so that we have more available traps for the small mammals to go into to really better understand the density of them.
The next question, do you accept ticks from out of state for a fee?
We do. So individuals can submit their ticks to us for testing for a fee. I think the testing that we do for free for residents of Pennsylvania is $50 if you're out of state resident.
Let's see. Says, hi, Nicole, you said you measured soil moisture. Has that not yet been included in the environmental factor plots?
No, so we just started implementing soil moisture collection this year. So when we are doing our tick dragging, we are now taking soil measurements at all of the subplots flags that we have out there. So essentially each plot, there's three sort of it's split up into thirds, and we're taking soil measurements at each one of those areas, and we are almost done our nymph drags now. I think we have about one more week, and then all of our sites will have completed 2 complete drags for the season. And we're going to look at seeing if that soil moisture is playing a role in activity as well as density of the ticks.
I did just put the Tick Lab website in the chat for anyone who's interested. I'm also going to, while you think of some more questions, I'm going to launch our post webinar quiz. We have some more questions. Steven says ransacking is what raccoons are best at.
Yeah, we have had beaver issues too. Some of our one of our sites, we had beavers steal our raccoon exclusion devices for their. Yeah, so it's been fun. There's been a lot of, I'm surprised no one's asked about challenges. There's been a lot of challenges, with such a large scale project of just animals coming through. I mean, bears just stealing our traps and destroying them. It's been fun.
I've got a couple more here. Which assay do you use to differentiate Anaplasma HA versus V1? And do you see many that fail to genotype altogether?
Yeah, so we're using a genotyping assay that differentiates between the two via, like, I think it's a 1 SNP difference. We do sometimes have a few that fail. We have a few that fail more so in the passive surveillance studies, just because the ticks come in all shapes and sizes. Our active surveillance, we don't have as many that fail the genotyping. Altogether, they're just cleaner samples, and they tend to have stronger calls in general, just because they're not associated with exposure drying out and things like that. The paper off the top of my head that we're using this assay from, I can't recall, but it's a 1 SNP difference, and it's basically a TACMAN genotyping assay.
Next question. Do you recommend testing the tick rather than blood testing after bitten?
I presume we're associating human blood testing. So in terms of testing the tick gives us a lot of important information of potentially exposure to a tick-borne illness. So we do know with human testing that the effectiveness, the sensitivity and specificity of that testing varies. And it can become challenging to have a positive blood test result, and it does take some time for you to potentially build up an immune response. So testing the tick can give you a little bit more information sooner if you've been exposed, and then it will help guide your physicians on if you do develop symptoms, because we know a lot of the symptoms for tick-borne illnesses are similar amongst themselves as well as amongst other tick-borne illnesses. I mean, there's been cases of individuals being told they have COVID-19, but they have a tick-borne illness. So it's challenging. So if you have a tick and you're lucky enough to spot the tick, pull the tick and remove it, I do recommend testing it, because then you can have some information of potentially what you may be exposed to if you do develop some symptoms.
Does that answer change for non-humans?
In terms of companion animals, it's the same. So, I mean, all blood testing is typically an immunological test. We don't circulate a lot of these tick-borne illnesses within the bloodstream of our systems. So if you try to do qPCR on the blood, you might detect things that are bloodborne, like Babesia microti, or white blood cells infections like anaplasmosis, but you might miss infections like Lyme disease. So it is, if you have the tick, you have a better understanding of overall potential disease exposure.
Next question. Can you share more about your main funding sources that enable you to test ticks for PA residents for free?
So we are funded through the state through funding with the Department of Health, the Pennsylvania Department of Health.
That helps. Do you have any more questions? Just give everybody a minute to think. Maybe not. Says thank you very much. Excellent study and presentation. So yes, yeah, thank you very much, Dr. Chinnici. I did include the Tick Lab website in the chat here, so if you're interested in learning more or come up with a question after leaving the webinar, that would be a great place to go. Are there any other resources you'd like to direct people towards?
Yeah, so ticklab.org is our website for our passive surveillance testing. We're working on getting a portion of our data for our active surveillance also up on there. You can view some more of our statistics if you go to ticklab.org/statistics, and then you can kind of see some more of the rates of diseases. So we do test, I think someone asked, do we test ticks from outside the state? We do. And so that information is also on our ticklab.org website. So if you're just interested on how many individuals from your state are sending in ticks and what they might be carrying, you can find that at ticklab.org/statistics. And then if you want to follow us on social media, we try to do a lot of creative information to get the public educated on some things that they can be aware of. And we try to be pretty vigilant on doing that consistently a few times a week so that we can keep the awareness of ticks sort of in people's minds every time they're going outside. So you can follow us at PA Tick Lab on Instagram or Facebook.
Media at PA Tick Lab. Oops. Awesome. Thank you so much, Dr. Chinnici, for presenting and for these resources. And thank you to everyone for joining us today. We will see you next month.
Thank you, everybody.
Thank you. Bye-bye.